Really delighted to once more to welcome you here for one of our innovation talks, which is for initiatives, consortia, researchers, and regulatory governmental agencies and ongoing work related to health data science, broadly termed, but really with the purpose to raise awareness of initiatives local and global related to life science, broadly defined. As long as we have health data and advanced analytics that are part of the topic, it can be featured here. So please bear that in mind if you are interested in participating. We really hope to foster interest and knowledge sharing about health data science innovation, not only in the greater Copenhagen area, but globally, and also for the ability for initiatives to reach out to one another. If you'd like to be featured in our talk series, feel free to reach out to us at Lars's email, larn@dkma.dk. And then we'll definitely be reaching out to you. A simple disclaimer around this event, presentations here are not endorsed by the Danish Medicines Agency, and it will not be possible to present a solely commercial offering. I'm very honored to be able to welcome SØREN BRUNAK here today for this session we will have. And the purpose here will be for SØREN to present his enormous work around multi-step disease and prescription trajectories in the understanding of human disease progression patterns and underlying molecular level etiologies. SØREN will present his approaches around these trajectories, and just so we're looking at here, SØREN and his team uses data covering 7 to 10 million patients from Denmark. If you know our demographics, you know that's not the size of our population. It's actually a little smaller, but it actually covers the past 20 to 40 years. And then condense that data into, of the millions of individuals, their trajectories into smaller sets of recurrent ones, really giving us a completely different understanding about subgroups and longitudinal phenotypes that basically can inform us about differential treatment designs of relevance to those individual patients. SØREN is truly a pioneer in the biomedical sciences through his innovation and the introduction of new computational strategies, currently at the Center for Protein Research at University of Copenhagen, previously also internationally renowned for the Center for Biological Sequence Analysis at DTU. And I think, SØREN, you have published close to 300 papers, international peer-reviewed scientific journals, including some of the top-profile ones. Really, really happy to have you here on this session today, and looking so much forward to your presentation.
Yeah, thank you very much for your kind introduction and for the invitation. Here, I will just share my screen, and it is really an honor also to speak in this program because we are interested also in feedback because we are not what I would call classical epidemiologists. I mean, we come more from the bioinformatics and the big data and the data science area. So what we have been working on for the past years, 10 years or so, is to look at diseases longitudinally and now also prescription trajectories, as you will hear about later. And in this way, we are sort of disease agnostic, and we are interested in the longitudinal patterns primarily. We also work on the raw patient record in many of our projects, so not just registry data, but of course this is easiest to get your hands on. Raw patient record data might be more complicated, and we will also return to that. So the idea is illustrated here. I mean, if we look into the Danish National Patient Registry, we will see all these ICD-10 codes for different patients, and we will see them in the ICD-10 system, and you see the color coding. That is one of the two color codings I will use today. And I mean, basically, we're interested in what is the systematics in these trajectories. If you follow people over 20 or 40 years, what is actually the patterns that we see, And we are also interested in trying to see whether we could redefine some disease patterns, because we are using the ICD system. It goes way back. But I mean, is the disease for this third female here, these 4 diagnoses in a row, is that a disease, or should it be broken down in a reductionistic way into the diagnosis? So we're also interested in looking at the etiology and seeing whether we actually could condense some of the codes into fewer trajectories that would represent diseases. And as I said, I mean, ICD goes way back, more than 100 years, and of course this will always be behind the disease development. We've seen this with Corona and COVID. Of course, these codes, they got introduced in Denmark very fast when WHO released them, but of course the codes are behind the sort of real development. And in ICD, as you also know, I mean, we got more and more codes. It started as a causes of death system, and then more and more codes came in over time, and soon we are looking to ICD-11, where there will maybe be more than 50,000 codes. So more and more fine grains. So our efforts is maybe also to try to see whether we can condense some of this. This is also a system that is used for payment in healthcare. So it doesn't necessarily reflect disease etiology and mechanistic aspects. But of course, the data foundation, our registry data for this type of trajectory work, where we of course have to interface different versions of ICD if we want to go 40 years. It's not because the Danes suddenly got much more ill in 94, it's because the ICD-10 had more codes in the Danish version. So most of the work we have done in the ICD-10 era since 94, and the first paper here illustrates how we see the different code breakdown for females and males, for inpatients, outpatients, and emergency room patients. You, for example, see all the pregnancy-related codes to the left for the females. You also see chapter 2, the brown chapter here, is kicking in earlier. But we basically ask this question. We have, say, 7 million individual trajectories. Which are the most frequent ones? Which are the sort of highways in the disease space when we go across all the diagnoses? And here we see ICD-10 level 3. Of course, you can also go more fine grained. But we developed an approach to look into the statistics of this. And we should, of course, also remember, also in the context of this talk, that many of the diseases people get, at least some of them, will be treatment provoked. If you get vaccinated, you might get some diseases you would not otherwise have gotten. If you get radiation therapy for your cancer, might be different in terms of what complications you get compared to if you have chemotherapy and so on. So disentangling what is just disease etiology and interaction between diseases and what is treatment provoked is also very important. This is also why you will see in this talk that I talk both about disease trajectories, but also about treatment or prescription trajectories, where we would like to follow the systematics in the drugs that patients receive. And of course, there's a lot of work also going on now for finding biomarkers that can sort of understand some of these longitudinal processes. And again, there is a big difference often between, for example, genetic signals that will give you a disease risk and those signals that actually determine how fast you progress. So progression variation is also important, and to try to separate that from the basic disease risk. But of course, a lot of environmental influence, including the drugs, on these trajectories when we go into disease space. So, as you also heard from Jesper, I mean, we're interested in mechanisms. I will not talk so much about them in this talk, but of course we would like to look at the diseases to the right and the comorbidities and how diseases combine. But we're interested in finding the molecular reasons and the networks behind the disease progression. And there I usually also say that with the Human Genome Project that started in '89, I mean, the hope was that we could go bottom up and say, now we look at some genetic variants to the left, and then we would be able to predict the diseases that an individual would get. And this has not been very successful. There's a lot of heterogeneity, and also how diseases combine, and what is actually progression patterns versus just disease co-occurrence. It's only in the more recent years that people have worked on that. So we need to get both sides of this figure in order. We need to prepare the longitudinal patterns to the right, and then we could look for biomarkers. This is the idea. But I will not talk much about that. I will show this other slide that I often also show in talks. It's not from our own work, but this illustrates how many human genes people thought the human genome would contain. So we have time on the x-axis, and we have the number of genes on the y-axis. And if you look down there at 1960, people thought we would have around 7 million genes, and that was before the discovery of exons and introns. So people thought human genes were short, like in bacteria. Now we know that they are longer, and they are much fewer, and they cover maybe 30–40% of the entire genome. But it was quite remarkable that even the back of the envelope calculations when the Human Genome Project started sort of estimated 150,000 genes and 100,000 genes. But as the Human Genome Project progressed, we sort of down at close to 20,000 genes. So we have much fewer genes than we had anticipated, and this, of course, also means that presumably many genes are involved in more than one disease, and we have a lot of pleiotropy that also relates to the disease co-occurrence that we see in patients and in the clinic. But back to the trajectories, I mean, it's quite simple what we want. We would like to stratify patients according to longitudinal patterns. So each dot here is a diagnosis, and we might have the same diagnosis to the right. It's actually from a paper on sepsis that I will not go through, but where we found 3 different routes towards sepsis that actually were widely different in terms of the risk of dying from sepsis. But that's the basic idea in our work. We would like to not sort of make case control in the sense that one diagnosis versus not that diagnosis, but try to find these longitudinal patterns. And as I also said, we can do this at the level of diagnosis. We can do it at the level of drugs and prescriptions. We're also making lab test trajectories, and we're also doing quite a bit of text mining on millions of patient records and try to see what we can find of longitudinal patterns there, and I will not talk so much about that. I will get back to the prescription trajectories, but first I will just show a few slides on the disease trajectories that we have done over the past couple of years. But of course, taking this to the prescription level, where we have analyzed more than 1 billion prescriptions in the Danish National Prescription Registry, is interesting. So first, I just touch a little bit on the disease trajectories. You already saw this one, but what we did. Now we sophisticated it somewhat, but what we did in this paper that we published in 2014 was to develop a quite simple approach that deals with the combinatorial explosion. Of course, when you have, say, 1,200 or 1,400 level 3 ICD-10 codes, then of course you have many possible trajectories. So we sort of made a quite simple statistical approach. Now it's a little bit more complicated, but that approach identifies pairs of diagnoses where you have a statistically significant direction. So you have more patients going from N40 to C61 than the other way around. So we make these linear trajectories. You see 4 of them here, and then we can also summarize those as a graph where we, for example, add the number of patients that follow one of these steps by the thickness of the area. So the basic idea is quite simple. And now we have actually made quite many papers. This is from unpublished work. We are working on depression, and you see in the middle depressive episode, and you see recurrent depressive episode in green later there. And of course, in this kind of analysis where you summarize a lot of linear trajectories, you see the sort of complexity of some of these phenotypes. But the idea is, and the hope is also that maybe the past can be used to predict where the patients will end up in terms of outcome, or whether this can be used to differentiate between the optimal treatment for subgroups in this particular disease area. Here's another one for epilepsy where we work with Gitte Moos at Rigshospitalet on discriminating between focal epilepsy and generalized epilepsy, and also do a lot of drug analysis in that context. We also last year, 2 years ago, published a paper on sex-specific disease trajectories, and if you're interested in that, I can refer you to this paper. So all these transition probability for hopping between different chapters in ICD, we tried to highlight all the differences for the diagnoses that both sexes can get. So no ovarian cancer or prostate cancer in this one, but I will not go into detail with that. I'll just skip this one. But I would like to point at a paper we published last year that releases a disease trajectory browser where you can interrogate the ICD-10 period data, so data from 7.2 million people. You basically plug in some codes, and then the tool will take all the statistically significant pairs out and stitch them together. And you can also enhance, crank up the relative risk, and then some of them will disappear. And you can quickly get an overview over how is the disease progression patterns sort of looking like in Denmark. So this is not, again, disease co-occurrence, just it's sort of statistically significant directional forward-going patterns, and you can check all kinds of details. So this is just summary level data, so there is no person sensitive data in the browser. But I would like to mention this tool, and I mean, you just choose a disease as linear trajectories for Down syndrome, but you can choose, and then you can see whether there are statistically significant directional patterns around the disease that you work on, and then you might compare to Swedish data or Norwegian data or UK data or American data. And of course, we should also remember that the genetics is not the same and the healthcare system is not the same, so we are not necessarily expecting that all longitudinal patterns will replicate. So I will go next here to some of the recent work we have done on polypharmacy and analyzing patterns in prescription. And we have both looked at dosages, and we have also looked at just the sort of order in which you get your prescriptions. But that's of course a growing problem, because we have more and more multimorbidities in people, and we would like to understand which drugs have drug-drug interactions and which can be given together. And polypharmacy has, at least in some cases, just been addressed by counting. You say it's bad with more than 6 or 11 or whatever drugs, but our ambition was to try to say, okay, these 11 drugs are okay together, but these 3 are not. And of course, this is also inherent in a lot of trial work and so on, but we tried to go into that using raw patient record data, where we would have the dosage information ready for analysis. So we have one project called the Big Time Health Project, where we actually have data from the 2 eastern Danish regions, where we have 2.7 million patients followed over 10 years from 2006 to 2016. So that's our data set where we have all the lab values, all the prescriptions, actually also permission to all the images and sort of the deep phenotype that you can get out of patient records. And for technical reasons, we have in this project where we look at dosages focused on a data set from the capital region only, where we have 1 million patients and around 25 million drug prescriptions, and you see how they are linked to the ICD chapters down here to the right, and you also see statistics on how the polypharmacy situation look like as a function of age, that is as expected and described previously in the literature. You can also look at before you go into analyzing how drugs are prescribed together, just to see what are the sort of number of co-medications and the number of patients, and you see the different ATC. And here, of course, I switched to ATC in my color coding. I think I left out the slide on that color coding, but this is again illustrating across the ATC groups how the prevalence of therapeutic groups is, and the more prevalent, the more it's co-administered. And this is from a paper that Christina Leal have made. She just defended her PhD, and the paper has been submitted and is in review. So what she really did was to get into sort of a quite complicated model for defining dosage changes. Of course, you have several drugs. In this case, you have 3 different drugs, and then some of them change dosage, and you see the color is changed, and the color is indicating the dosages. So she developed a model for how actually to follow all these dosage changes, and then later possibly seeing how these dosage changes correlate with known drug-drug interactions. And in this data set, 185 million treatment episodes were defined, and it led to, due to statistical reasons and also not sufficient monotherapy episodes. I mean, we couldn't look across all 1,500 drugs in the data set, but it ended up with 413, and then a lot of co-medication drugs, so a lot of pairs. I will skip some of the details in this, but the idea in her work here was to say, okay, we have an index drug, and then it is prescribed together with other drugs. And you see the statistics here from these 1 million patients. So you see a lot of index drugs, and then you see the ATC code breakdown to the right, and it's fairly homogeneous across the drugs, how many you have from the nervous system and how many you have from the cardiovascular system and so on. There's some variation, but when you look at drugs where the dosages are changing a lot. So the dosage of the index drug is changing a lot or changing in a statistically significant manner across the different ATC groups. You see much more variation, and there is a tendency, for example, for drugs to change a lot when they are co-medicated or co-prescribed with drugs from the same ATC group. So what came out of this was that this data set of 80,000 co-medication pairs went down to around 4,000. So these are the ones that Christina analyzed in more detail. So for example, I could maybe point here at other nervous system drugs here, NO7. You see then suddenly when co-prescribed with other group N drugs here, there's much more variation, and you can see the same down here for psychoanalytics, that suddenly the N, but there's of course a lot of details in that. So one of the results of this work was to try to see all those 4,000 pairs here that tend to be changing a lot in terms of dosage. How many of those would correspond to known drug-drug interactions, including drugs mentioned in the Danish interaction database, but also other databases. So we have actually now put together 25 different DDI databases, and in this work, 15 of them were used. And it was interesting that for these 4,000 pairs, 83% of them corresponded to known drug-drug interactions. So out of these 309 drugs that survived this analysis, 287 and then their co-medications corresponded to known DDIs. And then, of course, we have 17% left. And one possibility is that these are representing unknown, yet unknown drug-drug interactions. And so what Christina did here was to, for example, look at the patient volumes and actually correct, as you can see in the first box plot down here. I mean the 17% lower patient volume. Also, we did quite a lot of text mining of 15 million full text papers. So not just abstract, but actually full text papers. And they were co-mentioned, these co-medication pairs, less in the literature. They also had higher odds ratios. So this point in the direction that I mean, these are maybe unnoticed because of the lower patient volumes, and therefore they are observed less. But this is of course just speculation. But it sort of illustrates how an analysis of systematic dosage changes can point you in direction of potential undiscovered drug-drug interactions. But why did we do all this work? Also, it is of course because we want to look at the adverse events and the adverse drug reactions. So we did already some years ago publish a study on the St. Hans Mental Center, St. Hans patient records, where we actually mined all the adverse effects that are mentioned on the Danish SPCs. summary of product characteristics. So you see here increased weight. So this is of course in Danish here, but you see in yellow the adverse events, and sweating at night and so on. So we have this text mining approach that we actually described in a paper in Drug Safety some years ago, where we only used it for monotherapy. So we mined all these SBCs and pooled the 21,000 adverse drug reactions mentioned there from all the commercially available drugs in Denmark, and this workflow was then used to find possible adverse drug reactions mentioned in the text of the record. And then, of course, it has to fit that the drug is prescribed in that period, but also that you don't see the same symptoms before the prescription starts. But this was a workflow we made for monotherapy, and now we are changing that to the polypharmacy situation. And then it would be interesting to then characterize all these co-medication pairs according to the adverse drug reactions that we actually see in the records, because we have, of course, a problem worldwide that we are not having a systematic recording of many of the milder adverse drug reactions. They are hidden in the text, they are hidden indirectly and implicitly in the lab values and so on. So if we want to link the polypharmacy to the adverse drug reactions in the text, we need to go into the text. And of course, also if we would like to link this later to genetics, then we need to do this exercise of actually at the individual level, individual patient level, we need to extract the putative potential adverse events and then try to make the analysis. But we have not done that yet, and Christina didn't get to make that in her thesis, but this is the idea. So next I would like to also describe briefly some other unpublished work that we have done on the prescription registry that I mentioned in the beginning. So this work, of course, is carried out on the dosage change work is carried out on real patient records. So now we are moving back to a prescription registry. So we simply took the Danish prescription registry and analyzed nearly 25 years of data. These 1.1 billion observations and see how are people sort of moving around in the ATC space, and how does it look when you look at the entire registry. Of course, the registry has been used for a lot of analysis that have been more drug specific. Here we are just making a drug agnostic analysis of all the sort of correlations that we find, and again you can make statistics as you saw for the patient registry. You take the lower panel here. We see ATC group counts as a function of age. We say males and females, and you see we get a lot of drugs when we are babies or very young, and then it goes away, and then we see some differences that are well known between the males and females in terms of drug use. So again, we need to deal with this combinatorial explosion. So we break it down to drug pairs and make an analysis of how one drug increases the likelihood that you will get another drug. I skip some of these details, but of course you could analyze this from the perspective of drug pairs that are going across different ATC classes. You can also work with the same ATC class and maybe look into how people shift from first-line treatment to second-line treatment and third-line treatment, and maybe potentially find out that there will be groups of patients who actually should start on second-line treatment. So this is one of the aims in this project, and the general result is sort of illustrated here, quite boring in many ways. We take all these 1.1 billion prescriptions, and then we find out which are the statistically significant trajectories that we observe in this space. And if you set a threshold of 1,000 patients that should follow these trajectories, hopping from one drug group to another. Alejandro Orocho, who did this work, he found 9 million of them, and that's of course then a basis for more targeted or specific analysis. But this is one way to discriminate between what is just 1 billion trajectories and what is actually statistically significant longitudinal patterns in the prescriptions. And actually there was quite a compression when you go from 1 billion down to 9 million. But we in this paper we go into this issue of whether we can see whether it would be a good idea, for example, in hypertension, or whether we can spot the groups that maybe should start on second-line treatment or third-line treatment instead of first-line treatment. So we compare ACE inhibitors here to ARBs, and here you just see the trajectory network for this specific group. And I mean, what we discovered when you compare ACE to ARB, and you compare 4 groups. So here we have patients who actually stay on ACE. So we have on this figure here years after the first RAS prescription here, and you have the survival probability of the y-axis. When you compare these 4 groups, staying on ACE, staying on ARB, change to ARB from ACE, or the other way around, then there's quite a bit of a difference in the mortality. So there could be an idea in actually finding out, should actually some of these be prescribed in a different way, or should they be checked for possible change in the prescription. Actually, the numbers here for hypertension, of course, quite high, ACE 550,000 and so on. So the statistics is quite good. So we actually took this example and looked into the UK Biobank. And as you know, it's not 7.2 million people. It's 500,000 people. So here we are down to 50,000. But so the statistics is not as good, but we see exactly the same sort of ranking in the survival probabilities, and we also go into other examples, but this is the idea. And what we also do in the paper that I will not have time to go into is actually to turn this into a prediction approach or a treatment plan recommendation approach, where we take the prior trajectories of the patients. So in this case, it's patients treated with ACE and patients treated with ACE, and then change to ARB. So we can make sort of a patient like me approach, where we compare a new group, a new patient with these 4 groups, and then we can suggest what the patient should actually be recommended, and or what the physician should be recommended to recommend to the patient. Of course, this kind of approach should then go into a trial to see if it actually works. But in this retrospective analysis, we can actually see the same pattern in the Danish data and the UK Biobank data, and of course also in the UK Biobank data and also in the Danish data. We can go into genetics, but I will also not have time to go into that. But that's the idea, and this is of course then now GP data in the prescription registry. So you've seen one example for the hospital data, and also one example for the GP data, and of course, optimally they should be put together, and we also work. So I'll just mention here in the end one example where we more actively use these disease trajectory for prediction. I mean, you saw in the previous examples that we have a model where we use the prescription trajectory for prediction, and I didn't show you the results in detail, but it's actually quite successful. But it's not published yet. I mean, here we switched to intensive care data. That's really a big data sort of area. And in a paper that we published a few years ago, we tried to use the disease trajectory of a single patient for a mortality prediction of that particular patient. We are not so good in Denmark at actually using our registries in the treatment and the bedside situation. I think we could do much more of that. But the idea is quite simple. Here we want to predict survival, 30-day survival, for example. And then we take the disease trajectory and plug it into an old school neural network, very simple perceptron that I also used in the 80s in my work. So nothing sophisticated there. Trajectory to the left, and then lab values and vitals and so on to the right. So the data to the right, they come from a 24-hour data collection, and then of course the disease trajectory could come from 15 or 20 years. So here we are really aggregating timescale, and this is of course timescales, and this is of course interesting. What is most powerful, the data that you have after 24 hours following admission, or the trajectory? And so it's illustrated here. So you can do this in various ways. You can look at the history before admission, you can look at the history at admission, and you can look at the aggregated history. You could also look at the performance of the SAPS-2 score that normally is computed from these values that you have after 24 hours. And you can also look at Apache and so on, other survival scores. But it's quite interesting that in this simple analysis, if you take the blue one, that's SAPS-2. So that's the performance in terms of mortality prediction that you can get from these data, from the vitals and the lab values and so on, in this correlation coefficient that we use here, 0.325. So the mortality is very high in this type of patients. So this is why it's also harder to predict. But if you take the orange one, you see that is actually quite a bit better. So using the disease history at admission, you actually outperform what is known, what can be known after 24 hours. But when you put them together in the green, where you use all the data here, then of course you get the best of two worlds, and then you get a better prediction. And that's the point here. There's actually a lot of power in and predictive performance hidden in these disease trajectories. So they presumably can be used much more at the n=1 single patient level. So the statistics you see to the left is for all the patients, and I will not go through the data set. It was 12,000 ICU patients, a lot of detail in that. But if you, for example, break it down into sepsis patients and respiratory insufficiency patients, you see, for example, for sepsis, I mean, the SAPS-2 score is really bad, and the history at admission or before admission really outperforms SAPS-2. For respiratory patients, it's still being outperformed, but it's more powerful. So this sort of illustrates how these longitudinal trajectory data can be used for a single patient. You can also go into explaining features with these machine learning algorithms, which features are important, and not surprisingly for intensive care patients, age is very important, lengths of hospital stay and so on. There's a lot of details in this. We also found some new features here that I will not have time to describe, but if you, for example, have a high age, so that means orange dot up here, then of course it's not so strange that you are getting drawn towards non-survival. But then when you look at how the age interacts with other features, for example, metastatic cancer, then you will actually be drawn the other way. So there are some of these interactions that the machine learning approach picks up that also are very interesting. And also we found some new diagnoses there. But getting into explaining the network and what features are important is, of course, also important. What in the past was actually important. And I just flash a newer paper from last year where we're sort of not just making a prediction after 24 hours. This is Hans Christian Thorsen Meyer who did it sort of a more deep learning approach where there will be a prediction every hour. And actually, he can explain the network every hour, what features are drawing the patient towards death and what features are drawing the patient towards survival. Actually, this patient here will survive, but it again sort of illustrates how the longitudinal analysis can be done. So I will finish now and be happy to answer questions. I mean, I'm trying to look at longitudinal patterns in general, whether it's diagnosis, whether it's prescription, whether it's dosages, and of course I've not talked about lab value trajectories or text-based trajectories, but that's the idea in our work, and possibly also redefine some phenotypes as trajectories that could be more mechanistic. Maybe the 3 sepsis trajectories that you saw in the beginning of the talk, maybe they represent 3 different mechanisms, and maybe they should be treated also in different ways. And we are doing a lot of trajectory analysis in a large number of diseases where we try to find the subgroups that actually maybe reflect different etiologies or mixed etiologies. We worked, for example, in dementia, where we published a paper last year in Alzheimer and dementia, where we also could see that maybe many more patients than previously anticipated would have Alzheimer and vascular dementia at the same time. So competing etiologies for the same sort of phenotype in many ways. So we're interested in that. But of course, I'm not talked much here about diet, genetics, socio-economic data. We are working on smart meter data that also tell you something about how people behave and so on. So there are many data types that you can add to this type of longitudinal analysis. This is just the beginning, and I think many other groups are, of course, also working on these disease progression patterns using some of these thoughts that we also have been working on. So here's my group, and thanks to many of the names here and the funders. And again, thanks a lot for your attention here and for the invitation, and hope there will be time for answering some questions.
Well, thank you, Søren, for a magnificent walkthrough of some of the big data power that is being unleashed, and especially since all of this is happening within the past 10 years, it's just amazing to watch some of the results here. There were a couple of questions coming through that I just wanted to make sure that we do cover, and I can see a lot of applause coming through. It's really a tour de force, really appreciate it. And that is asking how change in treatment possibilities, new drugs and treatment regimes taking over 24 years of time. How is change in treatment possibilities, new drugs and treatment regimes taken in over 24 years of time? So how did you account for that over the 24 years? I mean, that's a lot of changes over those years, right?
That is a really good point. And if you look into diabetes, for example, if you take the ICD-10 period, of course, the treatment options were different. Now, I didn't present any work including that period here, but we actually have a project where we try to compare the trajectories that are statistically significant in the ICD-10 period and those from the ICD-10. So I mean, the point is really important, but you can also use it actively when you take GLP-1 and so on. How did it actually change the trajectory? So there's no doubt that new treatment possibilities are changing some of these statistically significant pairs, and even for, COVID-19, the temporal properties are of course different today than they were 1.5 years ago, and so on. So this is a good point, but I think you can still use the approach to compare different time periods and then also see the effect of new treatments. But it's more recently that we started working on the drugs, and now we will actually go into that question a little bit more.
Cool. And Paul is asking, are the clusters seen in sepsis patients regarding severity of outcome applicable, i.e. the 3 trajectories? And then he comes, which is, by the way, Paul is one of our magnificent statisticians here, and he points to the fact that statisticians are extremely wary of clustering. Maybe you can talk a little bit to that. I think we're getting into some of those classic debates. But
so now I didn't really present the paper. It's a paper from 2016, and actually the reference should have been out here. It's Mette Bæk, who actually did this work. She was wondering that if you looked into the literature, what was the impact of diabetes? You see the pink diagnoses here. This is endocrinology. And so some papers said that diabetes would increase your risk of dying from sepsis, and other papers said that it was the other way around. And actually what did come out of this paper was that actually by stratifying the patients in this way, so the middle one, actually these guys have diabetes, but they also have cancer, then she could sort of rationalize why the relative risks were so different in different papers. So I just recommend the 2016 scientific reports paper. It's actually now becoming more and more cited because people can see that these actually represent some information that is relevant in actionable sense. I guess intensivists, they know a lot about this, but here it's systematized, and you can look at the statistics and see whether you can find ways to improve it. I wouldn't say that it's perfect, but we're finding ways to improve it.
Fantastic. You did mention that you brought in the UK Biobank to reproduce some of the results. Have you done more work about reproducibility across borders, kind of validation of some of your findings? What does the landscape look like for that at the moment?
Yeah, what we see, I guess, is just get this. See the same pattern, right? And of course, we have had these data now for quite many years, and we work with data from the Danish blood donor study and also the Copenhagen Hospital Biobank, and we can see whether we can replicate. But I guess that's the key problem. We have some Nordic projects where we are funded to compare what we see in the genetic data from Denmark, in Norway or Iceland. And then I would also mention the 1 million genomes project that finally Denmark joined. That project, we had been an observer, and again in this project people thought that we would take all the Danish genomes and put them at some European supercomputer, but it is a federation approach where the genomes stay in Denmark, they stay in the National Genome Center, but then they can actually be analyzed in the same way if you get the right permission. So this idea of having a federation approach where you do not move data, but you move the algorithm around, that is at least security-wise and also legally feasible. So that's the development we see, because we all need replication of our results, and especially in a small country like Denmark. Even if we have more to be proud of now, I would say that at least for some years we were behind on the whole genome sequencing in Denmark in many ways, but it's looking better now, and we need these data sets from the other countries to see and check up on the patterns we see. And also in pharmacogenomics, I think that that is a low-hanging fruit, obviously. Sure. If you can get the side effects out and you can find the the variant that provoke that side effect, then it's very important to check it up against data.
I definitely do expect that also with European health data space, new technologies will have to be applied in order to access data in different countries, but also the ability where you may be able to go wide in certain countries and deep in other countries potentially could be complementary to each other, right? So you may not necessarily need genetics everywhere, but you would definitely need a lot of data in order to then go and look at some of those specific genetic patterns. I see Nana had a question. Nana, do you want to say that yourself? I think you raised your hand.
Yes, if it's possible, I would like to ask a
Please
question. Yeah, thank you for a very a great talk. So actually, I was interested in this figure that you showed because actually, you can see there's no difference in mortality whether you change from one RAS to another RAS. But actually, if you stay on the same treatment, your mortality is higher. So I was wondering whether do you have any data on, for example, healthcare seeking behavior, or because this could be a result of that, and also the data you showed about the intensive care unit, people patient getting referred to the intensive care unit, there's always a decision behind it from the intensive care specialist. So there might be other things than just the patient or the values or the disease of the patient disease that's dependent on whether they get to the intensive care unit, but also the decision by the physician himself or herself. So
these are good points, and of course this particular figure, if you take the Danish version of it, of course this is GP data. So we optimally would really need the GP records in order to see whether these would be patients who are checked up on less frequently or what is going on, and we also look to ydelsesregisteret now to follow up on some of these things. But of course we are not really in a good position, for example, text mining the GP records in Denmark, as you see, for example, in the UK. So we have some possibilities for following up and maybe see what is actually the either socio-economic or sort of follow-up aspect to this. Also very important, because this is, I wouldn't call it a confounding factor, but it's more possibly a cause, right? That they are not shifted over to the alternative treatment. But this is a really good point. And for the intensive care project, so this is a project done together with Anders Perner, so it's actually part of this Big Temp Health project where we had an intensive care component, and people, when we got the grant 5-6 years ago, people thought it was a little bit strange, but now I guess ICU data analysis has become a little bit more fashionable. But we go into some of these things because we couldn't publish the paper in Lancet Digital unless we replicated the result in a different region. So we got data from Region South. They used the same ICU database that was used in the capital region before SP. So there were some advantages there. So, but you can look in the second paper for some of these issues, and of course also Hans Christian. He's an intensivist and consultant in this area, so he actually started on his PhD after being specialist. So they are, of course, aware of all these decisions that are taken together with family and all that, and we comment on part of that in the paper. So I would say that. That this has been sort of incorporated in the approach, but of course there are confounding factors that you cannot necessarily exclude. But I think the idea of giving an explained network every hour gives the physician an opportunity to say, okay, now this feature seems to be much more important, so we change the treatment again. If it should be implemented, of course, there should be a trial, and this is what Anders Perner is expert in. So I will leave that part to him.
Thank you.
Well, Søren, thank you so much for truly a fast browse through 7 to 10 million Danes kind of healthcare data. I'm pretty sure we'll see a lot of more interesting stuff coming from you and your group. So really looking forward to that. Definitely food for thought of moving in some of the big data analytics also within the regulatory space. So thank you for the presentation. And thank you to all of you who joined in today to listen. And hope to see you around soon again.
Thanks. Thanks to you all also for the nice questions. Thank you.